genotyping data management for GenBank in this presentation. So we are not in April anymore, it should be May, but that's fine. And you will understand during the presentation why we are having this seminar, trying to connect that with what is being done in GRIN-Global Community Edition. So just as to give the context, over the past years, genomic has become more and more important in the management of GenBank accessions. So it becomes part now of, let's say, routine operation to characterize, to genotype the accession, to combine this information with phenotyping data. And this information has been very useful to correct the passport data of accession, to complete it, to look at the genetic structure of genetic resources, to look for redundancy of the accession. It's also a good way to look for, to make good gap analysis and to look for new accession to be acquired in the GenBanks. It's also a way to monitor the genetic integrity of the accession. Genomics can be also used for virus indexing as well. There are some techniques that are using this information to look for virus or pathogens in the accession. And it's also a key way to make some subset or core set in the collection. So this has been described in different publications. I just put this one that I found very interesting on the slide for the management of the GenBank. But we know that genomics can be also very important to promote the use of accession. So there are different ways to promote the use, but one of them is to promote accession from GenBanks for breeding programs. And as well, this type of example has been described in different publications. IPK in Germany were among the first one to make a very nice example of that on barley. But in the CGIAT, we have also a very good example of big project that generated a lot of genomic data from the GenBanks. And so I will refer to the 3000 RISE genomes that was published now a few years ago. That is a very important resource for the community. CIAT has been publishing also a lot of data, genotyping data on wheat with the mass of discovery project. And a bit more recently, CIAT has also published several thousand of accessions from the GenBank as well as breeding material to make a pangenome of chickpeas. So those are examples of what is being done with material of GenBanks and that are producing a lot of data. And since I work in the Alliance, I think it's also good to remind that it's for all the crops, maybe at different scales, but we are also producing a lot of data for banana, the ITC GenBank. Same in future seed, a lot of accession have been genotyped and some studies were published on cassava, for instance, to look at redundancy of accessions. So that's to give the context about the genotyping data, what has been produced, and now I want to talk a little bit about the data management side because it becomes easier to produce all this type of data. It produces a high volume of data, but storing those data, sharing those data, analyzing those data can be challenging. And 2-3 years ago, in the GenBank initiative, that is now the accelerator, it was decided to invest in a system that would enable to store the data and to share it with the community and for GenBank themselves as well, with the prospect to optimize the management of GenBanks, but also to enable further use with the user of the GenBank. So to look at, for instance, the structure of collections, to look at the gene context, to do GWAS analysis. Those are the type of examples that you can have with genomic information, genotyping data. So what was the landscape? The landscape of databases related to GenBank. So we have Genesis, of course, which is very well known for passport data. You have also now maps, you have traits, you have subset, but the purpose of Genesis was not really to deal with genetic data. So it was not an objective. Then we have GRIN-Global Community Edition. Very important for the management of the collection. There are a few fields where you could have some data, but the main purpose, again, and the design was not done to manage this type of information. Dataverse has been used quite often to store some data sets. So it's good to show that the data sets are public, can be downloaded, but it's not really a way to query the information. So it's like a static file that you download and you use on your computer. Then NCBI, which is the main place where you can deposit your data. So you don't have a lot of SNP data. You have most of the time over the raw data, and it's quite difficult to know what belongs to GenBanks, what belongs to the multilateral system, and what is coming from multiple sources. So it's a very generic database. Some centers develop also some systems that sometimes are quite specific. So SNP-seq was developed for rice in the 2000 rice genomes. It's a very nice system, but it's really tailored for the rice data, and the objective was not to develop it and to deploy it in multiple centers. Then we also have Germinate. Germinate is a very nice system developed by the James Hutton Institute. It has been used in several centers. It has been used in the BOLD project with Crop Trust. But here it's just to browse the data. You cannot really query the data. So it can be a good system, but it depends what type of analysis you want to do. And EBS was also investing in having a place for this type of data, but a few years ago it was working with the GOBI system and was not fully mature for genetic data. And GIGWA, that was that is co-developed, it's developed at CIAT and co-developed with the Alliance, was in place in ITC and also adopted by ICARDA. So this was the tool that we decided to use and to deploy as a test in the CGIA. So why GIGWA? It's because this is a system that is really able to manage huge amount of data. So most of the relational databases are not able to go at scale. When you import a gigabase pair of data, it becomes very difficult in MySQL, in PostgreSQL. So many systems have been trying that and are facing a lot of bottlenecks with that. So GIGWA is really able to deal with billions of markers. And it offers as well a lot of interfaces to interact with the data, to explore, to filter, to visualize, to export, and to share. So it was part of the expectation, I would say, in the community to interact with this type of information. In terms of technologies, so it works with Linux, Windows, or Mac. It's the database is MongoDB. That's why it's quite fast with big amount of data, and it's a Java framework for the interfaces. It's open source. The code is on GitHub. It can be deployed with a Docker application, and yeah, that's more or less what I can say about the technology. Behind that, if you have more questions after, we can discuss it. But it can be a very flexible system, so you can deploy it on a web server. So if you want to have a common access, and everyone can connect, and so you can invest in a powerful computer and have multiple users using a lot of databases or instance of GIGWA. But you can have it also locally on your own computer if you have small amount of data, not too big data. So it can be also very easy to use the system locally, and it's quite handy if you want to do training as well. So if you go in a place where you don't have a good internet connection, you can just have a local computer and show with the participant how to do it. If you invest in a server, there are different ways to promote connection. So you can create your password and login password for every user, but you can also include that in single sign-on. So for instance, CIAT has connected it to their LDAP system. So everyone has an account by default to connect. So as well, in terms of data management, there are a number of interfaces that are quite useful. If you want to, this is what you can see here. When you can manage different databases, so you can make some dumps of the data. So just in case there is a crash or you have a problem, you can restore the information quite easily, and you don't have to rely on IT to do that. So it's a good practice to do yourself your good management of the database and restore when you need before there is a more serious problem on a server, for instance. So that was just a short overview of what GIGWA is about. So after 2 years, we have been developing and deploying for the different GenBanks dedicated instances of the system. And so this is the home page where you can land, and for each of the logos, so you click on one of the logo and you will access one instance of GIGWA for the different GenBanks. So for some of them, it's in production. Some of them have data, but data are not public because it's still in the process to be analyzed. Some centers are testing it because they don't know if they are going to put it in place because there are policies like in CIAT that can be a bit more difficult for genomic data. But that's what you have online. So if you go to gigwa.cgir.org, you can access this and have a look to the different instances. So if you take one extensive like the ITC, you can have quickly an overview of the different projects that has generated data. So I will explain you a little bit how it works, but you create your data by dataset, by project, and everything is listed as a database where you have information about the number of variants and SNP markers in that case, and the number of individuals. And the ploidy, so it gives you an idea of what type of information you can find, and you, for each of these databases, I think what is important to say is that you can add metadata, and this is where we can make a link with information that is corresponding to the GenBank passport data. But not only, because the metadata part is very flexible and can allow you to add any information you like. The only thing is that you need to have an individual column where you have the same name as in your genetic data file. So that's the only requirement. But for the rest, it's quite flexible. What we have been promoting in the project is really to use the DOI for every data set that is submitted, because we want to be interoperable with the other systems. The DOI is like the common identifier that you can find in any system for GenBank in the CGIAT, and it's very important to keep at least this information to connect with other tools. So I know it's a bit conceptual, the slide at the moment, so I was thinking maybe just to stop for a moment the slide and to show you quickly what it is. So I was describing the portal, and so if you click on one of the logo, and this is in ITC in Leuven, so you arrive on this type of interface. So where you have just a description of the system, but what I was referring to is you can click here and have an overview of all the different data sets that are public. And I will take one. So we did some years ago a study on GWAS that was done with GBIAS data. So if I click here, I arrive on this type of interface where I can start browsing the genetic information. So I will have thousands of markers that are going to be listed very quickly on the system. And so this is what each line is a marker where you can see the position on chromosome 1, the start, the stop, the type of alleles you have, you have some genomic context. And if you click on one specific marker, you will have all the information on those markers for all the different individuals of your dataset. So basically, you have a huge matrix of accession, gene bank accession with markers. So it can be 1,000 by million of markers. So this is what the system is able to store and to display. And the added value of the system is that you can really query the information. This big file that is now in the database can be queried quite fastly by a number of fields here to filter. So you can select, I want only the SNP, and you are going to have only the SNP in your dataset. Then you want only the B allelic marker, and you filter again. You may want to take only on 2 chromosomes, you filter. So you see, it's quite fast to reply. You can work with groups, so you can work with your different individuals and select different individuals. So in that case, you're going to select only the marker for this type of individual. And this is where the metadata are important, because if you click on this magnifier, you have all the metadata that you want to put in your dataset. And I was saying you have the DOI, which is important for interoperability, but you have all the others that can be very useful to filter if you want, for instance, to select all the Musa acuminata. So in one click, you will only have the ones that are the wild type in your dataset. So that's a way to query your data, and then you have a lot of interfaces that can be useful for looking at the density of markers, for visualization as well. You can export in multiple formats. You can create, I think I've created one somewhere, just for the sake of time, because you have to download the data. It takes a bit of time just to download the file and visualize, but I have different databases that were created. So I think this one is loading, but this one is for ITC, but you have an instance for African rice here, where you have public data that were published recently for 9,000 accessions. And each time we can customize the interface with a logo, a different color. We can provide more information about the dataset with the person to contact. Is there any reference to the publication? Yeah, that's the type of information still loading. Okay. So I take this time just to show you what we have in IRI. So it's information that was extracted from a publication, and we could insert that on Napier grass, which is a forage crop. And what is interesting here is that we could integrate in the metadata a lot of traits, for instance. So people can make some selection of data based on different traits, and try to discriminate what is the genetic information for the different traits. Okay, still loading, but yeah, this one is for rice, where we put data very specifically for some genes. So I don't want to be too long, because I know that it's not the main purpose to explain what are the genetic data, but I think it's good to have a sense as well of how the interface are designed and what the user friendliness of the application. And if you have any question on the interface, we can discuss afterward, and I can go back on the application. But so it was what I wanted to say for the demonstration. But now, as a developer, I think it's important to know that this system is also accessible through an API, and it is compliant with the API, the breeding API, which is a standard way to extract data. And this is why it is very important, because the idea was not to have silo data for genotyping data. We want to be connected with the other system. And we know that by this way, by having the good metadata and by having data that are accessible to API, we can start designing use cases with connection with other systems. So if you go to GWA, you have a menu where you can access the documentation in Swagger, where you have all the data for the breeding API. You have all the calls that are documented, and then if you know how to do it, you can start designing this type of commands that you will have here, where you are going to query information for one particular DOI and to retrieve information. So you can, for instance, make a call on a specific DOI, and if the application replies to you, you will know that you have genetic data or not for this DOI. So this is where we can start designing some application routine that would enable to know what is available on genetic data. And so we don't have to ask GenBank manager what's the level of genotyping in your collection. There could be automatic way to get some statistics about that and to get the data if you want to get the data. So you can directly make this call in the interface in the Swagger, and you can have an idea of the output for particular accession. So this is what I did a call, for instance, on bin data at FutureSeed, where with a specific germplasm ID, I could get this information and the associated DOI. So those are just examples of what you can do with the application. And now, what is work in progress is to see what could be the integration of data. So that's something I've tried to design. I mean, there is nothing in stone, but trying to understand what's the data flow. So we have GRIN-Global that is providing data to Genesis. Genesis could be connected. I mean, GRIN-Global Community Edition, I think, can be or is compliant with BRAPI. Genesis is compliant also with BRAPI. Many systems are implementing this type of API. And this is where it becomes interesting to think about what could be designed. So we could imagine that some applications are going to make some call to know what is available for genotyping data. We can also, by this way, connect with the breeding data, because many systems are using API like BreedBase, IDP, EBS as well is compatible with BRAPI, and Germinate as well. And EBS actually is also using GWA in their backend to store genetic data. So there is a way to foster interoperability between the system. So what I was thinking is, for instance, if you're in Genesis, and now we have AI chatbot on Genesis, so you can ask questions. It could be, give me the accession of bananas that have associated genotyping data. At the moment, it doesn't work because you don't have this connection. But it could be possible to create some connections and to have some routines that are going to check, to cache this type, to put in cache this type of information, and to redirect the user to the good resources. So we could imagine that for specific accession, we could have additional information in the passport data where you could have a link redirecting to the GWA database where you can find genotyping data. So this, in theory, now is possible, and that could be investigated by trying to cross data together. It could be done as well with a subset, because you could also register subset for genotyping data. And we could also imagine some linkage with that, redirecting to the data on genetic data to be investigated as well. And we have been done that in MGIS, which is a Musa germplasm information system that we developed for bananas. So it's a website where we have multiple types of information, not only about the ITC, but it's released a lot of data about the ITC collection. And we have been creating facets here, a bit like in Genesis, where you can see what are the different genotyping studies. It redirects to pages where you have data on genetic data, and you can as well go to GWA and to access the dataset. So I think there are some brainstorming to do how it could be maybe done for all the gene banks. And I want to finish the presentation by talking about GRIN-Global Community Edition, because I think this is what you are interested in here in the community. But I think there are ways as well to do that. I think it's very important to track information when you extract. I mean, some accession are selected to make DNA extraction, and I'm talking here in the digital gene bank in FutureSeed, and in front of me I have the lab where a lot of those extractions are being done, and there is a system in place. I mean, to do this tracking, to have identifier for different tissues, to know exactly, I mean, to track what happened with the DNA extraction, to whom it was sent, who was the vendor, and so on. I know that there is a plan to have a more elaborated tracking system in GRIN-Global Community Edition. It's not done yet, but there is a plan to do that. And I think that's a good moment to think about what could be important to have in different systems to make this connection, to make sure that we can really link the physical resources that are linked to the accession, the DNA information, and how to link that to the information. And it's important for quality control, for accuracy of data and reliability. And at the moment, as I said, we have access to DOI in the data, but there are other type of information that could be stored in the metadata in GIGWA and maybe in GRIN-Global Community Edition as well. I don't know, and I think it will be open for discussion in the coming weeks, months, and happy to be part of the discussion for that when it's a good moment. So with that, I just want to close my presentation and open time for questions. Thank you very much for your attention, and happy to take any question.
Thank you, Mathieu. Thank you very much. And again, apologize for the link, but everything went well. So the floor is open, like I said in the beginning, because we didn't have the time really to present Mathieu, but I think we know very well his work, particularly in bioinformatics and genotyping. So I think it's a very, very interesting dialogue that we have to do. Well, how can we incorporate and how can we get more of the data, genotyping data, and consequently of the phenotypic data, and how to use that for the gene banks and for the plant breeding. So I also shared here some of the works where you can see the value, and some of them also mention or point out what Mathieu also present a little bit in a different way. And we are open for questions. So please, any question, doubts, comments, just raise your hand, just unmute yourself and ask. Mathieu, I think I will start until our colleagues think a little bit about it. Can you expand a little bit more, because some colleagues from the EBS, they are here. Mathieu from Genesis, they are also here. So how can this really be interlinked? And as I put here in the chat, next seminar, it will be or 28 or 29, and Khadija will also present a tool that was supported by ICARDA. And every institute is doing different tools according with their needs. How can these all be integrated? How you see it? I know that there is a lot of discussion on it and needs more discussion, but can you give an insight about it, please?
Yeah, well, that's not easy. That's my first answer, because we have different frameworks in the CGIAR. So you all know that there was a reform, and that we have different now science programs. So GenBank is an accelerator. So we are Breeding for Tomorrow, which is another science program. So it would be very easy to not discuss together, but I don't think it would be suitable to do it. So we need to find what are the common framework, what can be done. And I think this type of seminar is an excellent example where you are bringing people from multiple perspectives, and where you have an open forum to do it. And I think that's already a good start. Then I think there is the willingness to cross system together. It's very easy to stay focused on its own system and ignoring the rest. But at some point, we need to step back a little bit and see what is the global picture. And I think it's based also on the willingness of people to do this exercise, which is not easy because you have to go a bit outside of your comfort zone, because you have to understand the other system, the other constraint of the people, and to find common ground. So I don't have a silver bullet for that. I just share my perspective, which might be very different of the ABS perspective. I should have mentioned that a lot of effort has been done, and I have been part of it on the genotyping request for the breeding program. So a lot of thinking and conceptualization has been done, and we can for sure benefit from that in the gene bank as well. When it comes to implementation in system, this is where it has to be discussed. We have a lot of constraints, but I think the key word is interoperability. And I've tried to convey that with the API, but again, API is not as well the only solution, because if no one is designing a way to connect things, well, you have connection in theory, conceptually, but you don't have a system that is really functioning across the different use cases. And I don't know through the different framework what would be the best way to interact, but for sure we are open for this type of discussion in the framework of the gene banks. And if we're invited to do it in different frameworks, we will come and we will discuss and we see what are the possibilities.
Thank you, Mathieu. I don't see any hand raised.
So, hi, Anna. Can I ask some questions?
Yes, of course.
Anna, thank you very much for sharing the link. Yeah, faster. Yeah, thank you very much. So, Mathieu, how are you? This is Rajnish from IITA. Nice to see you, and nice to see this GWA. Really, this is useful because sometime I have published some paper. They ask, give me where is your data. So already I loaded the data in the GWA. I said this is our link. So data is here. This really is work for us, very easy for us, not to go like Dryad and fix here to load the data and give the link. Then we can load it our engine bank GWA and we can give that link to them. It works good. My question is this, Kuban. As you say, so that in the data, you have the SSR data, and now I'm sure we are in the new technology, we have the SNP data, am I right? Now there are two data set and accessor and original plasma is the same. Now, if somebody is accessing both the data, SSR data and this, and how he can compile and look, the lines was in the 10 years back in the SSR data, and the lines based on the data today on this comparative study, the line is the same or not? Is any facility there in this GIGBA? Are you planning to do that or not? Yeah, that's maybe tricky, but you can think on this. Because either we put one type data, this is here, so people will not compare. If they are going to compare and they are not getting anything, they will not go on the technical side for the reason, but they start to say the line is not look same and this, and but once it will start to spread, it will be faster than we convince them technically, you know. So that's is my one thread. And another is I request one thing. Suppose if I'm loading my data and somebody, can you make some technically like this? If somebody is looking my data or using my data for their research, at least I get the notice ki who is using this data. I mean, somebody downloaded, which institute downloaded, who was the person, because somebody is coming with some information to join GIGWA. So at least I know how many person has downloaded my data and if they are, and what the purpose they are using my data. I mean, that's my understanding. Because if you are putting the open access in something like this for the future. And third one is very important. Again, I'm asking my third question technically. Now, suppose I have done my cassava data for 3 years back. So that was the version genome 6. Now we have updated version 8, or maybe after 2 years, there's a pen genome. So do you have any facility immediately? We know that my version was 6. And can I know what will be my 6th version SNP data now located on the 8, and what will be the new SNP location of the chromosome on position, so that we don't need to go realignment technically, rather than we can move on this. If you can help on this, so it will be also helpful. So these are my just 3 comments. In the future, how can you add it if it is not available now?
Okay, thank you for the 3 questions. I'm going to try to reply them, and if I I misunderstood something, you can complement. But yeah, you have all the collection have a lot of legacy data from microsatellites, and I think they were able to manage it because it was a smaller scale and to put in some databases. GWA was designed at the moment where SNP data appeared, and it was not possible to manage it. So consequently, it's not the place where you store SSR markers, because it's not done for that. It's done for SNP data, indels. So now how to compare? Again, the system is done in a way that you can store your data and make some filtering, export, and make your analysis. So it's not a silver bullet, but you probably have a genetic tree, a diversity tree from your SSR markers. And from your STEAM data, it's quite straightforward to select what you want to extract as a marker, export it in different formats. It can be dissimilarity matrix. We can export that. You can be a FASTA format and make your own tree. And then you compare different trees and you see what in terms of diversity. You are not going to do that directly into GWA, but GWA is supposed to help you to make your own comparison quickly, because it's difficult to manage those big data sets. It's difficult to filter if you are not a bioinformatician, and you need to make a lot of conversion of formats. So this is what it is supposed to facilitate. It's not going to take your SSR markers, take your SNP marker, produce a tree alone and make comparisons. So it's not able to do that, but it should help you in this process. So that's for your first question. For the second question about data availability, I often have this question, and it's up to you. Let's say that you have made a publication. What happens most of the time, people do their publication, they have a file in Dataverse that can be accessible without or with login, it depends. And the data are already public. So why not putting the whole data set in GWA and make it public as well? So that's the philosophy for the public data. But if your data is not public yet, you can just put behind a login and provide the login to the people to access it. Now, to monitor who is accessing that, if it is open access, it's only the web analytics that's going to tell you it's coming from this country or this country, but we don't have names. And actually, with some legal constraint in Europe, you cannot track people. It's forbidden. So you can have an idea of the origin, but you cannot do that really. If you want to apply that, so you could decide that your data sets can be accessed only with login and password, and then you would know. Then you would know who can connect the data. So that's the second one. And for your third question, it was about
genome update. Genome update, like different
genome updates.
Like,
that's well, if you have a different technology, or if it can be not compatible, usually you create a new database in the system, and you can make some similar analysis and try to compare your data. If you have exactly the same technology, like you go for Dart and you have different batches, there are ways to increment. But you will have to do the basic analysis to do your mapping. If it is different technology, you have to do your mapping anyway. If you are several batches, you can increment in the database, but that's something we discussed in the past already, Rajnish, I remember. We would need a concrete example to make some tests with your data.
Mathieu, my issue is this, like suppose last year I updated with the version 6, I loaded it, yeah. Now I have my version 8 and something. Now I'm doing, so it means if I'm version 6 is there and I'm loading again version 8, so it means data is going to duplicate based on the just reference genome version, but the markers and everything is the same. So it means more burden is going on. And same my comments on this, if I'm doing one study genetic diversity, I publish for 200 lines. Now tomorrow I'm going to GWAS for disease, again I'm the same data line, I'm putting it, either something is this, so I'm not sure the duplication of data is going, and one time I'm going to my full data set again, Cassava. So I don't know, replicate will be too much, and the load, I don't know. That's my just simple. So maybe
well, that's probably something that we should discuss specifically together in a separate call and really look at the data and see what can be done. But thank you for your question.
Thank you.
I think we should go to Monica, because she raised her hand.
Chamba is first and after Monica. So Chamba, can you unmute yourself and ask, please.
Okay, yeah, thank you, Anna. Thank you, Mathieu, for this presentation and the demo. So I think you answered some of the questions my colleague Rajnish came up with. So, and looking, because you talk about the at which point we can make this data available and accessible. So now that there's a discussion going on DSI, so how do you see the integration of this genomic data so that we have a proper traceability of, and integrated in the benefit sharing?
Mm-hmm. I have been involved in, I'm also part-time with the policy unit, so I spent some time in the CBD at COP and also in the Plant Treaty, so the DSI, I know very well what is happening. After COP, there is a decision with a set of recommendations to be applied to databases, where basically they don't request to track. So there is no tracing and tracking for the data. You need to inform user about the existence of the multilateral fund that has been put in place. And you also need to request people that upload their data to certify that there's a permission to do so with regards to the origin of the material and the data. So it requires actually, that's something that is ongoing, to make a few modifications in the GGCE interface to be compliant with CBD. But still, we will have to see what happens in November after the governing body of the Plant Treaty, because they may add some conditions that we would have to take into consideration. But I think there is nothing that prevents people at the moment to share their data because it was recognized that it should be compliant with, consistent with open access. So open access is concerned, but you need to take into consideration a few principles that are the FAIR principle. It can be the CARE principle if you deal with indigenous data. And so that's what we are going to see how to translate that. But there is no big bottleneck to share the data. Having said that, being part of the CGIAR, we have probably an additional responsibility because we give access to large amount of data for multiple gene banks. And there is a working group that is being put in place for the CGIAR with a different data manager of DSI data. It's just starting, and there will be a reflection about what is best. Should we be totally open access without registration? Or should we apply maybe some level of control? There are pros and cons. CIMIT, for instance, would be in favor of that because they already have some policy in place with Dataverse where you have to register and to accept some conditions. Maybe it could be a bit lighter now, but yeah. So in theory, at the moment, we can be compliant with CBD very easily, but we can decide maybe to be a little bit cautious with the Plant Treaty discussion and to apply something else. So I don't have the definite answer for that. It's ongoing work at Jamba. Yeah.
Okay, thank you.
Monica, thank you for waiting. Please unmute yourself and ask to Mathieu.
Thank you, Anna, and thank you, Mathieu, for the presentation. I'm happy to have you here at Future Seeds and also to be able to contribute in the thinking of how to link the data or the processes that are generating the data that will be placed in GGCE. So I think my comment and my questions will in the direction of Jamba. So we just had a webinar where we learn about the updates of the multilateral system, the ABS and DSI. And my question is directed to that aspect. So we know that if Genesis is for requesting material and has information associated to the accessions, so users normally go and make their request, and through the SMTA, they access the physical material. So now that, as you propose, having like which accessions have genetic information, so people would probably make a click and go to GWA or any other source where the data is stored, and then will access the data. But then I don't know, it seems to me that it's been somehow disconnected. If there is going to be any SMTA through this access of genomic data, I'm always struggling to understand. And also, so we have seen that you show in some slides that different gene banks have already published their work on large-scale genotyping. So many of them and us have to make this data open available because for publishing is requested. And sometimes we are not sure if we're doing things right or wrong. But knowing that we are going, the ideal world is to have the genomic data connected with the passport data in a platform. So how are you considering GWA and other platforms that Dataverse that I know, NCBI doesn't work this way, but have any agreement of people accessing the data so that then this ABS in the future can at least be tracked down? So yeah, considering that the contribution now, if it's for commercial use, is not an obligation. I don't know. I don't know if my comments and questions are not very clear, but what will be the position for GWA to make this data available, and if there is any way that we can track people accessing the data, not limiting, but just tracking?
I would say that it's not up to GWA or to the team or to me to decide what it should be. What we have to do is to be compliant with the existing environment. And after long negotiation in the CBD, they reach an agreement where you have a list of things that can be implemented by the gene banks, and it would be compliant with the CBD. The CBD includes the agricultural sector at the moment. And what it means is that until the plant treaty reach an agreement on DSI, it's only the CBD that applies. So everything that's being discussed in the governing body at the moment is only negotiation. So nothing is in stone, and we don't have obligation as CGIAR. So it means that, and I'm not sure it can be done retroactively. So I would say that it has been open access for genetic data for a long time without any consideration of ABS. And you can find those data all over the place in NCBI and other databases. So now we are trying to structure our data for gene banks, and I think the first objective would be to be compliant with CBD. That's my perspective. But it will be up to this group in CGIAR to decide what they want at the end. And we will have to realign if there is new thing from the plant treaty. But we are in this strange situation, developing system in the gene banks, because anyone from any university can order material, sequence, put it in a database, public database elsewhere, and they don't have to take care of that. Not really. Now with CBD, they have to take care a little bit. But being in the CGIAR, we have this additional responsibility and this type of consideration that we have to. And I understand why you're asking the question. But I don't know myself if we have to be over cautious as long as there is no clear rules to manage that. That I don't know. But again, it's not up to me to decide. What I see that there has been a long time of practice for open access for genetic data. We have a little bit more restriction or recommendation that are not compulsory. I mean, that's on voluntary basis. But I think we should adopt them, of course, to be compliant in CBD, and then adopt what will be decided in the plant treaty. And just remind that those restrictions are for commercial use. So in the CBD. So it means that for academics and people that are not doing commercial use, you still have flexibility to use those data, open access and without too much restrictions. Yeah, I don't say that my answer is much clearer, but I hope it gives you interesting insights, Monica.
David, do you want to unmute yourself and ask?
Yeah, I'm sorry, but I'm also gonna ask about the access, because I understand that there's a discussion about policy, but in your demo you showed the API access to Swagger, to the GGCE hosted by Future Seeds. We need to request a token there. Is that token based on a passport authentication, or is it still open access, all this data on this platform?
Well, you have different cases. So we, I mean, in the case of the banana data, so we have been doing a lot of research over the past decades. So a lot of publication, and as all the publications, the data has been requested to be public. So it was in NCBI, it was in multiple places, and I don't see why it would not have been public in the GGCE instance we put in place. Again, if there are new restrictions in the future, we can change that and put the password, but our approach has been to make it open access. Then different gene banks can have different perspectives. And for Future Seed, most of the data are not public. And Monica agreed at some point to have one data set to be public on Cassava because it was published and the data were on Dataverse. So you have this information, but you have many of them that are just with login for analysis and for data sharing. And this is the case for many other gene banks. Africa Rice has made a publication as well. So data were also in Dataverse, in the institutional Dataverse. Raw data was NCBI, so why not having SNP data on GGCE when we are trying to structure that? And to facilitate a lot of process for the gene banks, as I mentioned, it can help for a lot of tasks.
It's not so much the data sets that I'm asking about. I'm really asking about the access to the API. Is there?
As the API is there is no. I mean, you can access it. Yeah, it's open access. Yeah.
Okay. All right.
Yeah, it's open access. Yeah.
Great.
Mathieu, I think Norma is not anymore in the meeting, but she make a comment and a question. Amazing tool to centralize the genotypic data of gene banks, beside filter for some quality factors and visualizing distribution of markers in the genome. Is it possible to do some phylogenetic trees, or what other analysis it's possible?
Yeah, I think I touch a little bit on that in my response to Rajnish, is that you don't do it directly in the application, but the application allows you to filter the set of markers you want to use for your analysis of phylogeny. And there are ways to convert in the appropriate format to do that. So you don't do it directly, but you are going to gain a lot of time to do it.
Out of the time, we were also a little bit like 7 minutes late. I don't know if you have any other question. If you don't have any other question, you can reach Mathieu for more information, even for supporting, which is and this week we will announce the next presentation that will be by Khadija, and my son that how to manage all these tools and how to put together everything in a very nice way so it can be useful for everybody. So I think there is no more question. Thank you very much, and see you in the next seminar with the link, hopefully. Thank you. Thank you very much, Mathieu. Thank you.
Thank you, Anna, for the invitation and everyone for coming and all the questions. Thank you very much.
Merci.
Thank you, Mathieu. Thank you, Anna.
Thank you.
Thank you.
Thank you.
Thank you,
Bye.
Anna. Thanks a lot. Bye.